Hi, it's Dr. Phoenix. Today we're going to be looking at cellular respiration. Up until this point, we've seen how plants can undergo photosynthesis and make their own food, and they are known as autotrophs because they have the ability to do that. Animals are called heterotrophs because they need to get their source of raw materials and energy from eating things, eating plants that make the energy, or eating other animals that ate the plants that made the energy. Now, a common misconception is that plants photosynthesize and animals undergo cellular respiration. But in fact, that's not the case. Plants do photosynthesize and can make their own food, but all cells undergo cellular respiration, even plant cells. Plants, after completing photosynthesis, have created glucose molecules, and that glucose still has to be converted into a different form of energy in order to be utilized by the cells, just like our cells have to break down glucose into a different form of energy. This process that we're going to be discussing, cellular respiration, occurs in the mitochondria of cells. If you recall from our study of cells, the nickname for the mitochondria is the powerhouse of the cell. And that is because this process, cellular respiration, which breaks down glucose into a usable form of energy for the cell, takes place here. Now, if we look at the mitochondria, we're going to see, similar to the chloroplast, that it has a double membrane. It has an outer membrane and an inner membrane. And this inner membrane ends up taking on folds like this. And the purpose of these folds is just to make more surface area available for this process to take place, this process called photosynthesis to take place. In between the outer and the inner membrane, there's a space called the intermembrane space. And inside the mitochondria, inside the inner membrane, is a fluid type material called the matrix. Okay, this would be analogous to the stroma in the chloroplast. Okay, the matrix. All right, so the definition of cellular respiration is the process that releases energy by breaking down glucose in the presence of oxygen. Oxygen is a key player in cellular respiration. Now, glucose itself is an energy molecule. It can be burned. 1 gram of glucose, for instance, C6H12O6, can be burned in the presence of oxygen, and it will release 4,000 calories of heat. The problem is, if our bodies utilized glucose as their energy source, we'd catch on fire. That is way too much energy for our cells to use for the activities that it puts on. So the energy in glucose has to be broken down into smaller packets of energy. And if you recall me talking about the $10 bill, glucose is a $10 bill, we need to break it down into quarters, which would be analogous to ATP. Alright, so for this reason we don't burn glucose. Instead, we gradually release the energy from glucose by converting it into ATP molecules. Alright, so we said that this takes place in the presence of oxygen, and it does. And when glucose is broken down in the presence of oxygen, we call this aerobic. We call it aerobic, and cellular respiration is an example of an aerobic condition. Now, it is possible to break down glucose, at least partially, without oxygen, and that would be called anaerobic. And an example of anaerobic conditions would be fermentation, and we'll talk about that near the end of today's video. So here's the equation for cellular respiration. 6 oxygen plus glucose is going to yield 6 carbon dioxide plus 6 water plus energy in the form of ATP. Now, if you look at this equation carefully, you'll see that it is photosynthesis in reverse, essentially. if you replaced ATP energy with sunlight energy, it would be photosynthesis in reverse. All right, so let's begin looking at cellular respiration and the breakdown of glucose. First of all, regardless of whether oxygen is present or not, the first stage of breaking down glucose is always going to be a process known as glycolysis. And in glycolysis, what happens is glucose, a 6-carbon molecule, is essentially split into 2 3-carbon molecules. And those 2 3-carbon molecules are called pyruvic acid. Pyruvic acid. In the process of doing this, some of the energy from glucose is going to be picked up and stored in the form of ATP. We're going to gain 2 ATP molecules from doing this. Also, some of the hydrogens and some electrons are going to be spilled out of glucose, and they're going to be picked up by a molecule known as NAD, N-A-D. And NAD is going to carry these electrons and hydrogen ions to a further step in this process, as we'll see in a moment. So glycolysis is the process in which 1 molecule of glucose is broken in half into 2 molecules of pyruvic acid, each a 3-carbon compound. Now, glycolysis takes place in the cytoplasm, whereas the other 2 steps of cellular respiration, the Krebs cycle and the electron transport chain, they do take place in the mitochondria. But glycolysis takes place outside the mitochondria in the cytoplasm. And glycolysis is very fast. It can produce thousands of ATP molecules in milliseconds. And it does not require oxygen for this to take place. The problem is it will soon run out of NAD, N-A-D, and it will stop if oxygen is not present. But fortunately, when oxygen is present, then this process can go on, and let's see exactly what's going to take place. All right, so again, glycolysis is the first stage of breaking down glucose. It occurs in the cytoplasm, and glucose is basically split in half into 2 pyruvic acid molecules, sometimes called pyruvate. The hydrogens and the electrons are going to be picked up by NAD and carried in the form of NADH, and they will be carried to a later stage of this process, and we'll talk about what happens later on. During the splitting of glucose, we're going to create 2 ATP molecules, which can be used as an energy source for the cell. Now, these pyruvic acid molecules are going to diffuse into the mitochondria, and if oxygen is present, the second stage of cellular respiration will take place, and that's called the citric acid cycle. It's also called the Krebs cycle, K-R-E-B, Krebs cycle. Either name is used, so know them both. In essence, what's happening, these pyruvic acid molecules, which are 3-carbon molecules, are going to be broken apart. The carbons and the oxygens are going to be split off, and they're going to be released as carbon dioxide molecules. So for every glucose molecule that enters this process, we're going to have 6 carbon dioxide molecules released. Again, there's going to be more electrons and more hydrogen ions that are going to be released and picked up by some more NAD and what's known as FAD, and these also will be carrying these hydrogens and electrons to the electron transport chain. During this process, we're going to create 2 more ATP molecules for a total of 4 so far. Now, all of these high energy electrons and hydrogen ions are going to be carried to this electron transport chain, and this is the final stage of cellular respiration, and this takes place in the mitochondria but on the inner membranes. As these electrons and hydrogen ions are coming in, oxygen is going to be used to actually capture these high energy electrons and hydrogen ions and will convert some of that into water. So the oxygen is going to grab some of these hydrogens and it's going to form water. But what we're going to see is there's going to be an accumulation of hydrogen ions on one side of this membrane. And just like in diffusion, when there's a high concentration on one side of the membrane, they're going to want to try and move across the membrane. Well, they can't move across the membrane except for one place, and that place is through this molecule called ATP synthase. And when these hydrogen ions pour through this molecule, it causes it to spin kind of like an egg beater. And it causes ADP and phosphates to blend together and ultimately create ATP. This process is going to create 32 ATP molecules. And so when it's all said and done, 2, 2, and 32, cellular respiration creates a total of 36 ATP molecules. Each of the 3 stages of cellular respiration then captures some of the available energy in glucose and produces ATP. Because oxygen is required in the final steps, the entire process is considered aerobic. Again, glycolysis produces just 2 ATP molecules. The Krebs cycle produces another 2. The electron transport system produces 32 for a total of 36 ATP molecules produced. Again, this assumes oxygen is present. I want to show you that I've again made a drag and drop activity for you to practice labeling this diagram of cellular respiration. Follow the paragraph. The first stage of cellular respiration is number 1, and that of course is glycolysis. And you can drag it to number 1. This takes place in the cytoplasm. You can drag that over to number 2. And I'm not going to complete this, but you can go through this and practice the stages of cellular respiration. And again, if you can do this and have it in your head, you'll be able to answer all the questions that I'm going to ask you about cellular respiration. So what happens if oxygen is not present? If, for instance, we are running around the track at a fast speed, we're going to find that in a little while we're going to start getting tired, we're going to start breathing heavy, and we may get to the point where we get so tired that we have to slow down or stop. And we're just not able to get enough oxygen in through our heavy breathing to continue aerobic respiration. So what happens is we switch over to an anaerobic form of respiration, and this is called fermentation. With fermentation, we're releasing energy from glucose without the presence of oxygen. And so there are 2 different kinds of fermentation depending on the type of cell we're talking about. Our body cells undergo what's known as lactic acid fermentation. And as I said in the beginning, glucose is the first step of breaking down glucose is always glycolysis, and that is this top section right here. Glucose is broken down into 2 pyruvic acid molecules. If you recall, I mentioned that NAD is going to pick up hydrogen and electrons to become NADH, but very soon NAD will be exhausted, and when that runs out, glucose is unable to dump off those hydrogen and electrons, and this process up here will stop. We saw that in aerobic respiration, the process was able to continue because pyruvic acid was able to go into the Krebs cycle and electron transport system and release the hydrogens and electrons back. But when there's not oxygen present in humans, pyruvate is broken down, or changed, I should say, into what's known as lactic acid. And this is called lactic acid fermentation. And the good thing about that is when pyruvic acid is broken down into lactic acid, NAD is regenerated. The electrons and the hydrogen ions are released, and we recreate more NAD, which enables more glucose to be broken down. And so we can have this glycolysis process continue to take place, at least for a period of time. What slows this down and stops it eventually is lactic acid is toxic to our muscles and will cause our muscles to start to hurt and tighten up and cramp up, and that's what's going to cause us to stop exercising eventually, is the buildup of lactic acid in our muscles. And once that happens, then we'll have to stop, and as we rest, then the lactic acid will slowly be reconverted back into pyruvic acid, which can then re-enter into the cellular respiration aerobic cycle. Now this happens again, as I said, in our muscle cells. It also happens in yogurt, for instance. The fermentation process of yogurt is lactic acid fermentation. The second type of fermentation is called alcoholic fermentation. And this is what goes on in making wine or beer, or even making bread. Again, we always start out with glycolysis. At the end, we end up with 2 pyruvic acid molecules. In alcoholic fermentation, what happens here is pyruvic acid is broken down. One of the carbons comes off of carbon dioxide, or one of the carbons comes off of pyruvic acid in the form of carbon dioxide. And then we're left with a 2-carbon molecule, which is going to be converted into ethanol, which is alcohol. So for instance, if we're baking bread, the breakdown of pyruvic acid is going to produce carbon dioxide, which causes the bread to rise, and alcohol, which would be cooked out when you bake the bread. If you're making wine, again, the carbon dioxide will diffuse out of the wine for the most part, and the ethanol or the alcohol will be used to ferment the wine. And the same thing with beer, but beer would capture the carbon dioxide as well for the carbonation. All right, so that covers cellular respiration, both aerobic cellular respiration and the anaerobic process known as fermentation. I hope that's helpful, and if you have any questions, please feel free to bring them to class, and we look forward to seeing you tomorrow. Have a great day.